Hello, everyone. So today I'm going to talk about the concept that I did not cover during the lesson. So to help you to better prepare for the examination. Now, for the first thing that I want to uh, go through with you, it will be the final factor that affecting the enzyme activity. But before I talk about the final factor, I just want to quickly remind with all of you about this the previous two factors that will affect the enzyme activity. Now, if we still remember that actually there are not only the temperature and also pH, okay, uh, for us to affect the enzyme activities, actually there are also some other factors that we can see in the lab or in the experiment. So for the first one, definitely the temperature will affect the uh, second. So first of all, the temperature will affect the enzyme activity. So if the temperature it is not suitable for the enzymes, for example, if it is at a very low temperature, then in this case the enzyme it will be inactive. Inactive meaning that the enzymes actually it would be working in a very slow pace. Okay, the reason is that because the kinetic energy is not sufficient enough for the enzymes and substrate molecules to be successfully collide with each other. So in this case, okay, they will be at a very low reactivities, okay, the enzymes. But then, okay, if it is too high, okay, what means if it's too high? That means it exceed, okay, the optimum temperature. So what means optimum temperature? Optimum temperature, you can think it is the temperature that it has the highest reactivities for the enzymes. So it will create, okay, the enzymatic activities with a maximum rate. So if it exceed, okay, the maximum rates, then in this case, it will be what we call the optimum temperature, or not exceed, but I mean, if it is the maximum rates, then in this case, it would be the optimum temperature. But then, okay, if okay the temperature okay further exceeds the optimum temperature, then in this case we will say that the temperature actually it will be too high. We will say that it is at a high temperature. So at a high temperature, the enzymatic activity it will be decreased. Okay, that is because the high temperature it will cause a change in shape of the active sites of the enzymes molecules. So once the active sites, the shape of the active site of the enzyme molecules has been changed, then in this case, we will say that that particular enzymes, it will be denatured. Okay, the denaturation of the enzymes or the um the change in the shape of the active sites, actually, it will cause the substrate no longer fits into the active site. So in this case, we will say that the enzyme substrate complex cannot be formed because of the substrate no longer fit into the active site. So therefore, the enzymes will lose the catalytic ability permanently. So in this case, we can see that one of the factors that affects, okay, the enzyme's activity, it would be what we call the temperature. Now, the second one, it would be the pH, okay? So the pH, it will be having similar things. So, for example, if you still remember that, okay, uh, so the, sorry, just a second. Okay, so the second one, it would be the pH, okay? So you can think in this way, okay? So one of the example, it would be the um, amylase. Now, for example, there are two types of amylase that you have learned. For example, the salivary amylase, okay? Then in this case, if it is salivary amylase, we notice that the pH value or the pH environment that's suitable for these type of enzymes to work the best or to work at a higher speed, then in this case, it would be what we call the pH 7. That means the neutral environment. But if, okay, for example, if the pH it is slightly lower or maybe it is extremely low, for example, maybe at pH 2, assume, or maybe it is very acidity, okay, then in this case, maybe it is pH, not acidity, I mean, if it is very alkalinity, meaning that it is very alkaline or the pH is very high, for example, maybe pH 12, then in this case, okay, there will be no any reaction at all. The reason is that because if it is deviates from the optimum pH or the pH that the activities of the enzymes work the best, then in this case, what happened? Then as the unsuitable pH, it will also cause the denature of the enzymes, meaning that the uh, the shape of the active site has been changed. So in this case, the substrate will no longer can fit into the active site of the enzymes to form what we call the enzyme substrate complex. So therefore, the enzymes will again lose its catalytic ability permanently. So in this case, we can see that if there is any deviation or the change in the pH environment, it will also cause the enzymes to lose its catalytic ability. Okay, so now, okay, then any other factors that will affect the activities, actually there are quite a few. Okay, so apart from the temperature, apart from the pH, okay, there is also some other factors that we have learned in the laboratory setting or in the lab that we have done with the MLA speed. One of it will be what we call the substrate concentration. Okay, so if it has a very high substrate concentration, then in this case, for example, you can think it like there is a lots of substrate. Then in this case, this will also cause a higher rate of a um, enzymatic reaction. But then, okay, apart from that, there is also, for example, the enzyme concentration. If there is a very high enzyme concentration, then in this case, okay, this will also increase the uh, enzymatic activities. For example, if you still remember from the lab, okay, actually, there is an increase in the amylase speed. Amylase speed. Okay, so if there is a high amount of amylase speed, then in this case, it shall have a faster or quicker, what we call the enzymatic reaction. Okay, now for the last one, it is very special. There is a presence of what we call the inhibitor. So what is inhibitor? So inhibitor, actually, you can think it like some substance that can decrease the activities of enzymes. So what is inhibitor? Okay, you can think like inhibitor is a substance that can kill, okay, the enzymes, but not necessarily it is kill, but by how? By like changing the shape of the active sites. So how can we do or why there will be have such inhibitors? Okay, for example, like what we call the cyanide. Now, the cyanide actually it is what we call a type of inhibitor. Now, this inhibitor you can think it is a substance that can um, prevent the enzymes to uh, function, uh, perform the function properly. So in this case, the enzymes, as it no longer can perform the functions by like changing the shape of the active site. So in this case, what happened? Um, it will cause, okay, the enzymes cannot, okay, bind with the substrate. Then this cause a decrease in the enzymatic activities. Now, there is also some other example, for example, like the heavy metals. What is heavy metals? Okay, for example, some very large sized metal ion, for example, the mercury ion, the copper ion, etc. So these are also a type of heavy metals, and they are also some kinds of inhibitor because they look like the same, or the size are very bulky. So in this case, they will stuck in the active site, or they are difficult to be removed. Okay, so maybe let me use a more systematical way to, or maybe a metaphor to explain, okay, the two different types of inhibitor. Now, uh, if you look at some DSE paper, or if you look at some like older version textbook, you will see there are two terms, okay, for us to describe inhibitor. The first one, we call it what we call the competitive. What we call the competitive inhibitor. The competitive inhibitor. So, and there is another type, we call it the non-competitive inhibitor. So what means, okay, competitive inhibitor and non-competitive inhibitor? Now, let me make a metaphor, or maybe let me use the chair as an example. Now, okay, this is the chair. Okay, very look like a chair. Okay, now then, okay, these chairs are actually similar to the enzyme. Okay, these are actually the enzymes, but it is the chair. Okay, now the chair is only fixed to some um, students to sit on it. Okay, only fit the 3 hour students to sit on it. For example, maybe like Jarif, or maybe Aditya, or maybe Nathan, okay, to sit on it. But now, okay, what happened? Okay, there is other people, okay, sit on it. For example, maybe me, okay. Now, in this case, I'm the person, okay, that sit on it, okay, so I occupy the chair. So in this case, can the 3 hour students still sit on it? Yes, it can, okay. So in this case, maybe they can only sit on the second, or the third, or the fourth, but I'm still occupying the first chair. So in this case, can they successfully do the reaction? No, because there is less enzymes is functioning properly. So we will call these, okay, as what we call the competitive inhibitor, because I'm trying to compete with you, fight against with you to occupy the uh, active sites of the enzymes. So therefore, okay, for example, the competitive inhibitor, it would be like the heavy metals, iron, or the what we call the um like, for example, like the methanol. Okay, now I'm not sure whether you heard about, there is in China, there is some news related to some thick wine. Okay, so rather than they use, okay, the ethanol, actually, it is one of the component of a wine that is suitable for human to drink. They change the ethanol into some what we call industrial alcohol, which is what we call the methanol. Now, for the methanol, actually, it is a type of alcohol that cannot be absorbed by our human. So this would be very dangerous because the methanol look alike with the ethanol. So once our liver do the detoxification, then in this case, sorry, we cannot do the detoxification. Why? Because the methanol will occupy the shape of the enzymes. Okay, I mean the active sites, the active sites of the enzyme. So in this case, this will cause the our liver cannot undergo what we call the detoxification. Okay, but now um, this is what we call the competitive inhibitor, a substance that try to compete with the original substance, which to decrease the activity of the enzymes. But then, okay, uh, there is another type, what we call the uh, inhibitor that can kill, the enzymes. But how? Now, assume, I am the person, who like very aggressive. So I don't want to let the 3 hour students, to sit on it. So what I do, I try to break the chair, okay. So once I break the chair, maybe I I'm a very aggressive person. So in this case, I break the chair. Okay. So I only left a half part of the chair here. So in this case, as the uh, chair is broken. Okay. So in this case, chair is broken. So this is very similar, to what we call the non-competitive inhibitor. For example, the cyanide or some like harmful substance, for example, like some mercury product, and or maybe some acid, These they are what we call the non-competitive inhibitor. These substance actually it will cause one thing, by what, So uh, they will be break, but not exactly break the enzymes, but they are trying to damage, trying to damage the active site, the shape of the active site, the shape of the active site of enzyme. Then in this case, the active sites no longer have the specific shape. So they cannot bind with the substrate. So these cause a big problem. So uh, the this will cause a decrease in the enzyme activities. Okay, so in the examination, we won't ask you the terms competitive inhibitor and also non-competitive inhibitor, but we will only ask you the inhibitor. But I just want to explain to you what means inhibitor. Okay, so this is just a brief idea related to what we call the inhibitor. Okay, now then, so apart from all these sorry. So apart from all these things, so actually we do have some other factors that may affect the inhibitor. Sorry, I mean, may affect the enzymatic activities. But these, 5 things are the major things that will affect the enzyme activities. So you just need to know that, whether it is optimum temperature, optimum pH, whether it has a very high substrate concentration, and whether it has a high enzyme concentration where there is no any inhibitor, then in this case, the activities, the enzymatic activity shall be the highest, if there is the optimum temperature, optimum pH, the high substrate concentration, the high enzyme concentration, and also there is no any inhibitor to be present, Now then, that is what we call application of our enzymes. So now this is to something that mainly basically you just need to memorize, Uh, but uh, this is not really the major parts that you need to know or you need to work on it during the uniform test, But just for your reference, But uh, uh, this is also one of the coverage. But to let you know, okay? But just it is not the major things that we would like to testify to, okay? Okay, now for enzymes, actually, okay, we use the enzymes every day, okay. Here are some areas or some parts, okay, that we will use the enzymes, okay. For examples, okay, we can see that, okay, we got some cheese, okay. When we try to make the cheese, we will try to add, okay, the protease, okay. The protease will help us to extract, okay, uh, the milk, okay, to help to use to coagulate, okay, the milk to produce the cheese. And apart from that, when we try to extract the juice from the fruits, okay, then we will also add some enzymes into it so that we can break the wall, okay, the plant cell wall. So that in this case, this can help us to increase the yield of the juice for us to be extract, okay? Then, okay, for example, maybe some environmental protection area, then we will add some what we call the cellulase, okay, it is a type of enzymes to be used in what we call to produce the bioethanol. Now, most of the fuels or most of the bioethanol, actually, it is a type of ethanol that produced from what we call some crops or some sugar cane or some coins, okay? Then in this case, okay, the enzymes will be added in the process when we are making the bioethanol. So in this case, the biofuels, okay, actually is mainly using the bioethanol as the fuels. So in this case, when once we add the enzymes, then in this case, the yield of the bioethanol or the production of the bioethanol will be increased, okay? So next, okay, for example, maybe we will also add some plastic, okay, maybe some enzymes, okay, into the plastic, okay, so that in this case, the enzymes can help to break down the plastic, and it is also why we can have what we call the biodegradable plastic uh, that can we can produce or manufacture. This because inside this plastic there is some enzymes, okay, and once okay they reach a suitable environment, then in this case they will start the degrading. Okay, so most of the time when we see the plastic, actually there are some what we call some long chain carbon atom. So if you learn chemistry, you'll notice that once there is a long chain of carbon atom, then in this case it will be not easy to be degrade or degrade from the I mean, it is very difficult for the long chain carbon to be degraded because of there are some bonding, okay, causing them stick together firmly for nearly a decade, okay. So once we got these enzymes, then it can be easily to break down into some smaller carbon items or some smaller carbon atom, okay. I mean smaller chains of carbon atom. Okay, so apart from that, okay, there are also some other area that we will use, okay, enzymes, for example, like cooking, detergent, maybe textile, medicines, etc. These are some enzymes, okay, that basically we will use, okay, maybe to clean our clothes, to make some meats, okay, tenderizer, or maybe to help us to clean our skins, okay, then these are some example or some enzymes that you may see, okay, for us to use, okay, in this daily life, okay. So we can see that actually, okay, based on the product or based on these things, okay, we can see that there are some properties of the enzymes. So the enzymes can speed up the chemical reaction, and this also shorten the production time, and it allows some mass production of the product. Many enzymes, they will be at what we call the moderate conditions. So in this case, okay, uh, this can help us to prevent some extreme conditions, for example, such as some high temperature or pressure is required in the production process. So this can lower the production cost. Next, the enzymes are specific in action. So in this case, if there is some unwanted reaction, then there will be not catalyzed, and those unwanted product will not be produced. Okay, so few of it they will be produced. Next, the enzymes are reusable. So in this case, this will lower the cost of the production. But also there are also some limitation. For example, the enzymes are sensitive to the temperature and also pH change. So therefore, enzymes cannot be used if the production process involves some high temperature or unsuitable, or maybe there is a quick change in pH, okay, throughout the process. And if the enzymes are easily affected by the inhibitor, so therefore, if we use, okay, the heavy metals, iron, then in this case, it may not be suitable for us to involve the enzymes in the production process. So therefore, we can see that, okay, that is how enzymes, okay, used in our daily lives. So this is the whole part of the chapter 4. Okay, so I hope you can understand most of the part, because chapter 4 is a very important part in DSE. Okay, so please let me know if you have any other questions, and feel free to ask me if you have any question. Okay, and today's lecture will be stop here, and thank you very much for watching.